Very good morning to all my dear students. Welcome to our Zoology online class. Hope you are very happy. In the last class, we studied about the fifth lesson, molecular genetics, and in that class, we studied about two important topics. One is the structure of operon, and the second one is DNA fingerprinting technique. Okay, so now I repeat the meaning of this word operon. Okay, the definition for the word operon here. Operon means a cluster of genes or a group of genes doing same functions or related function is called operon. Okay, and in this section we studied about the structure of lac operon also. Okay, here lac operon means in Escherichia coli bacteria, the lactose metabolism. For the lactose metabolism, there are three enzymes are needed. Okay, so with the help of these three enzymes only, the lactose metabolism is activated inside the Escherichia coli bacteria. So here this this is called, this operon is called lac operon. And finally we studied about the DNA fingerprinting techniques and the applications or the uses of this DNA fingerprinting techniques. Okay, my dear students. So today we can complete this fifth lesson. Okay, we can complete this molecular genetics lesson today. Okay, so listen very carefully. Today we are going to study about only one topic. So the name of our topic is HGP, Human Genome Project. Human Genome Project. So listen very carefully, students. Some 5 mark questions, 3 mark questions, 2 mark questions, and many 1 mark questions also there. So listen very carefully. See, Human Genome Project means it is a mega project. Okay, mega project means this project was first introduced in the world in the year of 1990. Okay, the Human Genome Project was first introduced in the year of 1990, and there are 13 years. Okay, so to complete this Human Genome Project, it took 13 years, more than 13 years. Okay, so that only some people saying it's a mega project. Okay, then, and this Human Genome Project is 25 times larger than the other organisms project. Okay, then next point. See, the human genome consists of 3 into 10 raised to 9 base pairs. So just imagine 3 into 10 raised to 9 base pairs are present in our genome, in human genome. Okay, then this Human Genome Project is closely related or associated with another one field. Name of that field is called bioinformatics. Okay, so here bioinformatics is one of the important branch of the biological field. So there is a relationship or there is a connection is present between this Human Genome Project and this bioinformatics field. Okay, my dear students, so that's all about this introduction about this Human Genome Project. So once again repeat all the points about this Human Genome Project. The first point is this Human Genome Project was introduced in the year of 1990, and it's a mega project because it took more than 13 years to complete. Then this project is 25 times larger than the other organisms projects. Okay, the next point, the totally 3 into 10 raised to 9 base pairs are present in our genome, and this Human Genome Project is closely associated with another one field which is called bioinformatics. Okay, okay, my dear students. Okay, say now we are going to study about one important 3 mark question. Say, goals of this Human Genome Project or the aims of this Human Genome Project. The first aim, there are more than 4 or 5 aims are mentioned in your book. The first aim of this Human Genome Project is identify all the genes. That means our genome consists of more than 30,000 genes. Okay, so our DNA is our human genome is made up of more than 30,000 genes. So the first aim or the first goal of the Human Genome Project is identify all these 30,000 genes. Then the second main aim or the second main goal is There are 3 billion chemical base pairs are present in our DNA. So, the second aim is determine all these 3 billion chemical base pair that make up a human DNA. So, the second aim or second goal is determine all the 3 billion chemical base pair that make up a human DNA. Then, the third main goal or the aim of this Human Genome Project is store all the genetic information in database, particularly computerized database. Store all the information related to the human genome in database. Okay, so this is the third aim. Then the fourth aim of the Human Genome Project is improve the tools for data analysis. The genome of the human beings is analyzed by using different methods, different techniques. So by using this Human Genome Project, the tools of this analysis is improved. Then the next goal is transfer all the related technologies to other fields such as industries. So the genetic information of this Human Genome Project is also transfer, or these techniques also related techniques also transfer to another field such as industries. And the last goal is same. This Human Genome Project, when the scientists prepared this Human Genome Project, they were faced many problems, many issues, ethical issues, legal issues, and also some social issues. So these issues are handled properly. Okay, so we have to handle all this or address all these social issues, ethical issues, and all these issues. Okay, so these are the some of the aims or goals of the Human Genome Project. So once again repeat all the aims. The first aim is identify all the 30,000 genes of the human. Second aim is 3 billion base pairs are present in chemical base pairs are present in human genome. So determine all the 3 billion chemical base pairs that make up a DNA. Then the third one is store all these informations of the human genome in database. Then the fourth one is improve the tools for data analysis. Okay. Fifth one is transfer these related technologies to other fields such as industries. And the last one is address the ethical, social issues, okay, and legal issues properly. So these are some of the goals or aim of the Human Genome Project. Hope you understood. This is a very important 3-mark question. Then methodology. Okay, see, now we are going to study about the methods of this Human Genome Project. Methodology of Human Genome Project. See, the Human Genome Project consists of 2 major approaches. Listen very carefully. The next 5-mark question. Okay, methods of Human Genome Project. The Human Genome Project consists of 2 important or 2 major approaches. The first approach is, this approach focused on identifying all the genes that express as RNA. So the first approach of this Human Genome Project is focused on identifying all the genes that are expressed as RNA. So this is the first approach of this Human Genome Project. And the second approach is sequence annotation. Listen very carefully. Here sequence annotation meaning the DNA is in this method, okay, in this approach, the DNA is completely isolated from a cell. Then the DNA is converted or break down into many small sized fragments. Okay, many small sized fragments. Okay, then by using suitable host and by using suitable vector organism, many cloned copies of these fragments are taken. So here in sequence annotation, first the DNA is separated from the cell or isolated from the cell. Then it is converted or break down into many small sized fragments. These the cloned copies or the copies. Okay, the copies of these fragments is taken by using proper host organism and vector organisms. Okay, then here maximum in this human genome project. Okay, in this method, in this sequence annotation method, two organisms are used as a vector. The first one is bacteria and the second one is called yeast. Okay, so here bacterial vectors are called bacterial artificial chromosome, BAC. Okay, BAC. And in some sequence annotation method, yeast is used as a vector. So it is called YAC. Here YAC means yeast artificial chromosome. BAC means bacterial artificial chromosome. YAC means yeast artificial chromosome. Okay. So by using this vector, many copies or many cloned copies of this fragment is taken. Understand? Then next step. Say. Then after that, after taking many copies, these fragments are sequenced by using some devices. Particularly in this field, one device is used. It's called automated DNA sequencer. Okay, so by using this automated DNA sequencer, the fragments, DNA fragments are sequenced. Okay, then the next step, the sequence informations. Okay, the sequence informations are stored in by using some computer-led programs. Okay, listen the last 2 points carefully. The fragments, DNA fragments are sequenced by using automated DNA sequencer devices. Then these sequenced fragments are arranged or stored by using some computer programs. Okay, by using some computer programs. Then listen the point, next point very carefully. Say genetic and physical map on a genome. Okay, genetic or physical map on a genome is assigned by using a restriction endonuclease recognition site and a repeated DNA sequences. So the genetic map, okay, listen very carefully. The genetic map or the physical map, okay, on a genome, okay, physical map on a genome is assigned by using restriction endonuclease recognition site or by using repeated DNA sequences. Okay, here repeated DNA sequences otherwise called microsatellites. Okay, otherwise called microsatellites. Okay, by using these two substances, these genetic maps is what assigned on a genome. Understand? Hope you understood. Okay. See, nowadays, okay, instead of using this automated DNA sequencer, some advanced techniques are used. One of the very important and very familiar advanced technique is shotgun sequencing. Okay, shotgun sequencing. So it is one of the, so that means by using some supercomputer, the fragments of the DNA is sequenced. Okay, so name of this the technique is called shotgun sequencing. Understand? And this method replace all the traditional methods. Okay, replace all the traditional methods we used once upon a time. Okay, so that's all about this methodologies of the human genome project. So once again I repeat this methodology of human genome project. Say this project consists of two important approaches. The first approach is focused on identifying all the genome. Okay, focused on identifying all the gene that are expressed as RNA. And the second approach is called sequence annotation. Okay, sequence annotation. In sequence annotation, what they are doing? First, the total DNA is isolated from the cell. Okay, isolated from the cell. Then the DNA molecules are converted into small sized fragments. Then these fragments copies are cloned or taken by using proper host and vectors. Here some technique bacteria are used as a vector, in some technique yeast is used as a vector. Okay, if it is bacteria it is called BAC, bacterial artificial chromosome. If yeast are used in this technique it is called YAC, yeast artificial chromosome. Understand? Then after that, these sequence fragments, the fragments or DNA fragments are sequenced by using automated DNA sequencer devices. Then after the sequencing process is over, all these informations are stored by using some computer programs. And the last point I said about this genetic and the physical map on a genome assigned by using restriction endonuclease restriction site or by using repeated DNA. Here this repeated DNA sequence is called microsatellite. And nowadays, instead of using this automated DNA sequencer, a shotgun sequencing, it's an advanced technique. Okay, by using some supercomputer, this technique is performed. So this technique replaces all the traditional or the old methods. Okay, my students. So now we have completed the methodologies of Human Genome Project. Okay, so aim of Human Genome Project over. Then methodology of Human Genome Project over. Then the salient features of Human Genome Project. Salient features of Human Genome Project. See, there are 10 points are there, 10 characters are explained in your book. Listen very carefully. The first salient features of Human Genome Project is, see, our human genome consists of 3 billion nucleotide base. Okay, there are more than 3 billion nucleotide base are present in human genome. Okay, only 5% of the genome only code for protein. Only 5% of the genome only code for protein. So this is the first salient features of Human Genome Project. Then the second salient feature is the largest gene's name is called dystrophin. Okay, so dystrophin is the largest genes of the human genome. Understand? Here, this dystrophin consists of 2.4 million bases. So totally more than 2.4 million bases are present in the dystrophin gene. So this gene is considered as the largest gene. Then, so this is the second salient features of the Human Genome Project. Then the third salient features of the Human Genome Project is, you all know there are 46 chromosomes are present, arranged in 23 pairs. The 19th chromosome consists of high density of genes. That means more numbers of genes are present in the 19th chromosome. Okay. Then the 13th chromosome and the Y chromosome consist of very low number of genes. So high density gene is present in the 19th chromosome and the low density gene is present in the 13th and Y chromosome. Hope you understood these third salient features. Then fourth one is, all the genes are showing variations. We can see large number of differences in the human genome. Then fourth, nearly 35,000 to 40,000 genes are present. Okay, 35,000 to 40,000 genes are present. Okay, and 99.9% of bases are similar in all the human beings. Listen very carefully. 35,000 to 40,000 genes are there. 99.9% of the bases are similar or same in all the human beings. Okay, understand? Then next, the sixth salient features. Say 50% of the identified genes functions is not discovered still now. Okay, so 50% of the known genes functions is unknown. Okay, just imagine 100 genes are discovered means 50 genes functions still we don't know. That's the meaning. Understand? Then the seventh salient features of this Human Genome Project. See, the repeated sequence Okay, the repeated sequence occupying the very large portion in Human Genome Project. Repeated sequence means the last class I told you. Some sequences are again repeated for several times. Okay, so these repeated sequence only occupying the larger portion of a human genome. Okay, then this repeated sequence directly will not code for any function. Directly they will not involve in any function, but they are playing a very important role in the structure of chromosome, functions of chromosomes, and the evolutions of chromosomes. So once I repeat this point, the repeated sequence occupying very large portion in the human genome, they will not directly code for any functions, but they are playing a very important role in the structure of chromosome, functions of chromosome, and evolution of chromosomes. Hope you understood this point. Then the next point, the 8th point is the first chromosome. Okay, today 46 chromosomes are there. The first chromosome consists of 2968 genes. Okay, 2900 Okay, 2968 genes are present in the first chromosome. Okay, then the Y chromosome, already I told you the 13th chromosome and the Y chromosomes only consist of very less number of genes. Already I told you. Here, the Y chromosome consists of only 231 genes. Hope you understood this point. Then the last point about the last salient features of the Human Genome Project is say scientists already identified 1.4 million locations of chromosomes where a single base difference is occurred. Listen very carefully. Already identified 1.4 million locations. Okay, locations of chromosomes. So in these locations, single base pair differences is occur. Here the single base pair difference is called single nucleotide polymorphism. Single nucleotide polymorphism. So the single nucleotide polymorphism is used to identify the locations of the chromosomes which are responsible for the diseases. Listen very carefully. Scientists already identified 1.4 million locations where the single gene problem is there. Okay, single gene differences is there, is occur. Okay, so these single gene differences or these single genes variations are called single nucleotide polymorphism. So this, what is the use of this single nucleotide polymorphism? This is helps to identify the locations of the chromosome. What type of chromosome? Chromosomes are responsible for the diseases. Hope you understood this last salient features. Okay, so now we completed one very important 5-mark question, salient features of Human Genome Project. Salient features of Human Genome Project. Okay, so once I repeat, the first one is there are 3 billion nucleotide bases are present in the Human Genome Project, and the second one is, okay, first one more point is there in the first point. Among these 3 billion base pair, only 5% of the genome only code for protein. Then second point about this human genome salient features is, say, the largest gene is called dystrophin. It consists of 2.4 million bases. Then the next one is the chromosome 19 consists of more number of genes, and the chromosome 13 and the Y chromosome consists of very less number of genes. Okay, very less number of genes. Then all the genes showing more variations or showing some differences. Okay, the next, see, there are more than 35,000 to 40,000 genes are present, but 99.9% of the bases are similar in all the human beings. Okay, then next one is 50% of the genes function is still we don't know. Okay, this is the next salient features there. Next, see, the repeated sequence. Okay, the repeated sequence occupying a very large portion in the human genome, but they are not directly encoded for any function, but they are playing a very important role in the structure of chromosome, functions of chromosome, and also in the evolution of chromosome. And the last one, scientists already identified 1.4 million locations where single gene differences occur. Okay, so it is called single gene polymorphism, single nucleotide polymorphism. The single nucleotide polymorphism is used to identify the location of the chromosomes which are responsible for the diseases. Okay, so my dear students, I hope you understood this question answer. Then next one is application. Applications of Human Genome Project. Listen very carefully. Human Genome Project playing a very important role in biological field. Now we are going to study about one by one. So this Human Genome Project is used to identify the genetic abnormalities. First use. The second use is this Human Genome Project is used to diagnose different types of genetic diseases. So genetic abnormalities can be identified, diseases can be identified with the help of the Human Genome Project. Then, say, if any any fetus are affected with genetic diseases, by using this Human Genome Project, the doctors can give counseling to the parents, okay, about these genetic diseases. So, with the help of this Human Genome Project, we can give counseling to the parents, those who are having children with some diseases. So, this is the next use of applications. Then, this Human Genome Project playing a very important role in another one technique, it is called gene therapy. Gene therapy means the process of replacing the damaged gene by a healthy gene is called gene therapy. So this Human Genome Project is also involved in this gene therapy technique. So this is one of the important applications or uses of the Human Genome Project. Then next one is this Human Genome Project is used to understand human biology. Human biology means anatomy, okay, anatomy, morphology, or everything we can understand with the help of this Human Genome Project. Then next one. This Human Genome Project also used to understand the non-human organism. That means other organisms. Other organisms. Used to understand the other organisms. Then next, this Human Genome Project playing a very important role in the healthcare field. Okay, healthcare field, agricultural field, environmental field, and also in the medical field. In medical field, this Human Genome Project is used to design different types of medicine to treat some symptoms and diseases. Okay, so now we completed the applications or the uses of the Human Genome Project. So one by one repeat all these applications. The first application is it is used to identify the genetic abnormalities. It is used to identify the disease or used in the disease diagnosis field. The next one, it is used to give counseling to the parents, those who are having children with the genetic diseases. Then it is used in the gene therapy field. It is used to understand the human biology. It is used to understand the non-human organism, and it is used in the health department, agricultural department, and then environmental department, and also in the medical field to discover different varieties of medicine to cure the diseases. Understand, my dear students? Understand? Then finally, we are going to study about the challenging of this Human Genome Project. Human Genome Project's challenges. What are the challenges this Human Genome Project faces? The first, there are three challenges explained above. The first challenge is, say, if the genetic sequence of a human being is easily identified by using this Human Genome Project, some people can use this information for profit and also for political power. So this is the first challenge. Okay, so let's repeat this point. With the help of this Human Genome Project, we can easily identify the genetic information. We can easily get the genetic information. So some people can use this genetic information for profit and also for the political power. So this is the first challenge. And the second challenge is, say, insurance, related to insurance companies. Sometimes because of the genetic risk, the insurance companies will not give the insurance money to the affected people. Okay, so it is indirectly it protect and it save the insurance company, this human genome project. And the third challenge is, say, breed out. Breed out means there is a fear is there. In futures, sometime with the help of this human genome project, okay, some superior quality human beings may be created by some scientists. Okay, so superior races of human beings can be created in futures with the help of this human genome project. So these are the three challenges this facing this human genome project. Okay, my dear students. So I hope you all understood today's class. Now we have completed the fifth lesson, molecular genetics. So today we studied some 3-mark questions. 3-mark questions and the aims of human genome project is a 3-mark question. Then the applications of human genome project is a 3-mark question. Then the challenge of this human genome project is a 3-mark question. Then the methodology and the salient features of human genome projects. These two questions are 5-mark questions. Okay, the number of 1-mark questions also there. For example, 3 billion base pairs, nucleotide base. Okay, and which chromosome consists of large number of genes and which chromosome consists of less number of genes. Okay, like that the 1-mark question may be asked. Okay, so today it's a very easy topic. So try to complete today and send the answer to me. I will correct it. Next class we can start the next lesson. Okay, evaluation. Okay, thank you. See you in the next class.